right, you ready to learn about cellular replication? As we search for signatures, in here, binary fission, 1 becomes 2, the history of cells, and the special case of meiosis. Let's begin. Replication. When studying the work of Gregor Mendel and the chemical basis of life, you learned that genetic material is used to store and pass on information in organisms. In genetic blueprints, you learned that material is deoxyribonucleic acid, or DNA. What does it mean to pass on information? In this lesson, you'll begin to take a closer look into how that happens. So our learning objectives today are to be able to describe the process and outcomes of mitosis, define the general steps of meiosis, its outcomes and its relationship to sexual reproduction, compare the outcomes and roles of mitosis and meiosis, and contrast the process of mitosis and binary fission. Apply concepts of genetic information and mutation to evolution. So we'll take a look back at our unit on evolution and how meiosis can create variation and mitosis can create more cells. So I like to say here, to keep this all straight, mitosis in my toes, meiosis in my babies. And so we'll figure out what that means here through this lesson. Here we go. Replication. You might think of how children look like their parents. Their shared characteristics are coded in the DNA passed down by the parent. This DNA is in all cells, which copies itself as one cell divides into 2. Do you think this process is the same for all cells? And we get to make a guess here. You can say yes, because all cells are the same. And hopefully we don't select that one because we know from our previous lessons that there's different cell types. Yes, because all DNA is the same, and we know that DNA has different nucleotide bases. Those can be different. So no, because prokaryotic cells would need more complex processes due to the cell wall. I don't know, that doesn't sound quite right. No, because eukaryotic cells would need more complex processes due to their size and complexity. Or no, because each species' cells must replicate differently. Okay, on second thought, I went back on all DNA is the same. So like DNA is the general structure of transferring or carrying information, we'll say. All organisms have DNA that does that. That can be true. Okay, binary fission. Prokaryotes being smaller and less complex than eukaryotes. And here you can dissect this word, pro meaning before. Karyo means cell or seed. So before the seed being the nucleus, before the nucleus. Being smaller and less complex than eukaryotes, U meaning true. Replicate using a simple process called binary fission. Listed below are a few steps which must happen during the process and a few that don't. See if you can identify which steps should occur in binary fission below. So if we take a look to the right, we have a cell, has its circular chromosome, and then it gets replicated, and now it has 2 circular chromosomes. And then the chromosomes are put to the opposite side of the cells, and then under cytokinesis, we have 2 different cells. So which of these is correct? DNA is copied in place. Sure. New cell wall is made. We don't know about that yet. Well, maybe here, that's a new cell wall. Cell wall and membrane pinch closed. That's true. They pinch here. The nucleus dissolves, and this is a prokaryote, remember, so we don't have a nucleus. DNA migrates to either side of the cell. We can see that happen. New DNA is made on either side of the cell. That's not true. Organelles replicate, and here it's a prokaryote, so we don't have organelles. Or 2 genetically identical cells are present. And that would happen at the end. So yes, let's see if we got that right. All right. So those are the steps, but what order did they occur in? So let's look at the order. So let's see if we can order these. Maybe starting with a 1 cell with DNA, and then this DNA replicates into 2, and then it moves to opposite sides of the cell. The cell wall and cell membrane start to pinch together, and finally after cytokinesis, they move apart. So I'm going to take a moment here and take some notes on that and write that down in my notebook. Oops. Okay, so here's how I set up my note. I put a little key. Most things in biology are circular, so you gotta label it. The green stuff is DNA. Here it replicates, so what are you gonna draw there? And then it moves to either side of the cell. And then this next step here, the cell wall starts to split, and then it eventually forms two cells in a process called cytokinesis. All right, so let's continue. Now we can answer these questions. Binary fission is used by prokaryotes and produces two genetically identical cells. All right, so that's correct, but what about eukaryotes? Eukaryotic replication. Plants, animals, and fungi are examples of eukaryotic organisms. They reproduce differently than prokaryotes. Which of the following do you think contributes most to this different process? Is it the presence of a nucleus? That's kind of the main difference here, so that feels pretty right. Different DNA, it's all the same DNA. Eukaryotes are multicellular, that is true, so that could be a factor here, but we can only choose one. So I'm gonna go through all these, but presence of organelles, eukaryotic cells do. Eukaryotic cells generally have larger genomes. That is generally true. There's this really cool, prokaryotes generally have larger genomes, different membrane structure, absence of a cell wall. Remember, there are certain types of eukaryotes that have cell walls. What type would those be? Those would be like your plant cells. Prokaryotes are larger than eukaryotes, or eukaryotes are larger than prokaryotes. That would more so be right, but I think presence of a nucleus, that's your best bet. And so yeah, as you will see, several of those answer choices are correct. Pay attention to how cellular anatomy plays a role in the process as you watch this next video showing a eukaryotic cell divide. All right, let's see it happen. Whoa, it looks super real, but I'm pretty sure that's a computer model. Okay, there's this little dance, the chromosomes get pulled to either side. And notice all the organelles as they get divided out to each different cell. And then what happens at the last step there so that the DNA can be held in a safe little structure called the nucleus. So we got a lot more things happening. Let's figure that out. All right, interphase, also known as G1, S, and G2. And then we have prophase, metaphase, anaphase, telophase, and cytokinesis. Those are the stages that we're going to go through. And we're going to take notes because that's a lot of new information. So here it says, normally the cell is in a state called interphase, which has three stages, G1, S, and G2, shown to the right. In interphase, the cell is performing normal functions, but towards the end, some changes begin happening. What do you see happening in the images to the right? So I see that in the left, there is one blue swirly bit, that's the DNA, and one red swirly bit, that's the other chromosome. So we have two different chromosomes. And then over here, I see that there are two of each type. So what must have had to happen between these two? Here, that one seems right. The genetic material is being copied. And let's take a look at this. Yeah, what do you think is happening in the arrows? So here is a cell in interphase, and here's another cell in interphase, but starting to leave interphase, so during G2, the genetic material is starting to condense. I think that answer probably would be right too. Okay. All right, so that was interphase. I'm gonna pause and take some notes there. Okay, so I set up each of my boxes, and then I took notes here. G1, S, and G2, the DNA replicates and the chromosomes condense. And then I wrote a note. This is the state that cells are normally, not really normally, but most of the time found in this stage. This is the most of their life cycle is spent just growing and doing their thing, making proteins, breaking down glucose, making energy, all the stuff that happens in a cell. That's the normal interphase. So then we're going to move into prophase, metaphase, anaphase, telophase, and cytokinesis. This is the process of dividing one cell into two for eukaryotes. So here we go. Now we are in, let's go to the next one. Prophase. The first stage of mitosis is called prophase. The images to the right show a cell both before and after prophase. What appears different? Okay, so here's before, and then there is after. So I see these little spindly things have moved to the opposite sides of the cell, and then the nucleus, I can see that starting to dissolve, and I can see my chromosomes now. So the cell has rolled on its side. Nope, there is more genetic material, not necessarily. There's less, not necessarily again. The genetic material has condensed, that's true. So it's usually just not condensed, it's just free floating within the nucleus. And then it actually gets into these nifty little forms where, see how it's an X? This can be easily pulled apart by what we call our spindle fibers. Okay, so it says, in prophase, the genetic material condenses from chromatin to chromosomes. So the state it's in, where it's just in the nucleus, is called chromatin, and it moves to chromosomes. In fact, while most biology students think of DNA in cells as the typical karyogram with X-shaped chromosomes paired up, it's normally in a less dense state. Okay, let's pause there, and I want to show you this because it's really cool. So karyogram, they use this word. If you ever don't know what they're saying, you can just Google it, right? But this is what you can visualize. All of our DNA is held in these. This is the condensed version. So there's chromosome number 1 all the way up to number 22, and then we have the 2 sex chromosomes that determine whether or not we're male or female. So if you're XY, you're going to be a male. If you're XX, you're going to be a female. And then, for example, just point out something interesting. Sometimes when you have 3 chromosomes accidentally in the third one, we call that trisomy 21. That's like Down syndrome. So sometimes when these pieces of DNA are different than this like normal state of the karyogram, that's when diseases can happen. For example, if you're missing one whole piece of number 1, you can see that that would be really problematic because all of this is coding regions, or a lot of it, for making genes that make proteins. Like, let's say this is, I don't know if it's necessarily true, but for an eye cell, if you needed to make some cones and some rods, that would be genes that are then encoded. Okay, back to it. Shown on, so now we're in that was prophase. Let's draw that in our notes and then answer this question. Shown on the right is an enlarged version of the diagram of a cell after prophase, in which X-shaped structures called chromosomes have condensed from chromatin. Do you think chromosomes would always be shaped like an X? Why or why not? So one of your answers could be yes, DNA is DNA. You could say yes, the X is due to the double helix, and that's maybe a tempting answer, but back to the karyogram. The double helix is it's actually even within the chromosome. So if we break down what a chromosome, so if I say genes to chromosome, might pull up. Okay, here we go. So it's kind of like this. So here's all my base pairs that we just learned about. All of those twist together to form the double helix, and then these actually twist again to form the chromosome. So it's all bundled up, and that's so you can carry many instructions together. There's lots of instructions for how to build a whole human. Oops. Okay, here we go. So then we could go, no, other organisms might have differently shaped chromosomes, or no, this DNA is different from the cell's normal DNA. Okay, so the correct answer here is that no, other organisms might have differently shaped chromosomes, like remember the bacteria cell that has a circular chromosome. And then I also find it interesting, so if you do a karyogram for a dog, what do you notice is different here? Do they have just 22 and then sex chromosomes? They have 38 different chromosomes. What do you think that's like for a monkey? Which are closer related to a human? Let's see. Looks like karyogram for a rhesus monkey. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. So yeah, here if you compare the human chromosome with the chimpanzee chromosome, you can see they're mostly the same, except for this 2A and 2B are a little bit different. That's pretty interesting. And yeah, I wonder how that evolved. It's pretty crazy. All right, back to it. So metaphase. Shown at the right is a diagram of a cell in the second phase of mitosis, or metaphase. What has happened in metaphase? The chromosomes are lining up in the middle of the cell. That looks right. The chromosomes have multiplied. That already happened in interphase. The chromosomes have connected to something on either side of the cell. Yeah, we have those little spindle fibers there. The nucleus has dissolved, but not in metaphase. The cell has not yet divided. Oh, I guess the nucleus has fully dissolved now that we're in metaphase. Prophase, it was starting to dissolve. There we go. A lot happens in metaphase. Geneticists often describe the dissolution of the nucleus as prometaphase. Well, that gives an answer. And the lining up, attaching of the chromosomes as metaphase. Okay, so I'm going to pause and take some notes here. Okay, so now we are in anaphase. It's the third phase. What do you think is happening? The cell is divided, not yet. The chromosomes have split in half. They have. The nucleus has dissolved already. The new nucleus has formed, not yet. And the chromosomes are moving to either side of the cell. That is true. Okay, I'm going to take some notes on that. Well, let's go to the next stage too. What is present on either end of the cell at the end of interphase? Anaphase. 2 complete sets of genetic information, 1 complete set of genetic information, half or no. So remember how it replicated in interphase? So we're still going to have 1 complete set on either side. Moving on to telophase, the 4th and final stage of mitosis. What appears to be occurring in telophase to the image at the right? Note, the pinching of the cytoplasm is part of cytokinesis, not telophase. It is shown here because 2 processes may occur simultaneously. Okay, so the cell has divided, almost. That's going to be that last stage of cytokinesis. The chromosomes have split in half already. The nucleus has dissolved already. A new nucleus has started to form on either side. Yeah, I start to see that. That's that turquoise part. The chromosomes are moving to either side. I already did that. And the cytoplasm is beginning to pinch inward. Yeah, we can see that happening. Oh, but they had just told us that's the pinching of the cytoplasm. Wow, so that is part of the next phase. Okay, I'm going to take some notes. All right, and our final step here is cytokinesis. After mitosis, 1 final process remains called cytokinesis. What happened in cytokinesis? Select all that applied. The cell has divided. Woo! Finally, we have a divided cell. The chromosomes have returned to chromatin. That is also true. The chromosomes, remember that's the uncondensed version. The chromosomes have split. Already happened, already happened, already happened. And that is the final step. The cytoplasm has been sealed between the 2 new cells. Oh, this has already happened. Okay, so just the cell dividing and the cytoplasm. All right, I'm gonna take some notes. Okay, in summary, mitosis is used by prokaryotes, eukaryotes, animals, plants, fungi, all organisms, so we could say eukaryotes, and produces 2 genetically identical cells. Woo! Okay, organelle replication. Although eukaryotic cells replicate using mitosis, mitochondria and chloroplasts are 2 organelles that use binary fission. Both of these organelles replicate on their own, not necessarily during the phases of mitosis. They are using the same method as prokaryotes. Thinking back to what you have learned about cell types, how are prokaryotes different from eukaryotes? Okay, prokaryotes usually have more DNA, not necessarily. Prokaryotes have more than one nucleus. They actually don't have any nucleus. They are larger. Nope, usually eukaryotes are larger. Prokaryotes do not have organelles. Prokaryotes have less DNA. Prokaryotes are smaller, usually. They do not have a nucleus. And that should be it. Okay, generally simpler organisms, they have less DNA. So there are 4 options. Okay, so the fascinating thing about the mitochondria is that there is this theory that the mitochondria was a bacteria that was actually engulfed by a larger cell. And so what is true about that smaller mitochondria is that it also does not have a nucleus, because it's kind of like a bacterial cell. It's like a prokaryote that's been engulfed by this larger eukaryote. No organelles, and an overall size smaller than eukaryotes. So this cell would actually, so there you go. We're going to talk more about that right here. Endosymbiosis. So if we break down this word, endo meaning in, symbiosis is living together, living in, symbio meaning sim, together. Okay, were you surprised by the similarities between bacteria and the cells, organelles, mitochondria, and the chloroplasts? Many early scientists certainly were, and there were several competing hypotheses for why mitochondria had the traits of bacteria and replicated separately from the cell, like bacteria. The leading hypothesis has become a theory commonly referred to as endosymbiosis. In short, endosymbiotic theory states that mitochondria and chloroplasts were once free-living bacteria that were taken in by an ancient organism in a symbiotic relationship, which we learned about that in our other lessons called mutualism. Research has dated this event to approximately 1.5 billion years ago. You learned about symbiotic relationships during your exploration in the Galapagos. Which of the following is a good description of a mutualistic relationship? Is it that both organisms benefit, one organism benefits and the other is harmed, one benefits and the other is not harmed? That would be commensalism. This other one here, one benefits and the other is harmed, is parasitism. Both are harmed, or neither organism benefits nor is harmed. So here would be both organisms benefit. Oh, both organisms are harmed. Yeah, okay. Okay, here we go down here. What benefit does the cell get from the mitochondria or chloroplasts? Defense, nutrients, genetic diversity, structure, or increased energy. Those are the places where cell respiration happens in the mitochondria, and then photosynthesis happens in the chloroplasts, and both of those are involved in our energy reactions. Okay, so they say here 1.5 billion years ago, this is the ancestor of plants and algae. When a cyanobacteria was engulfed, that became plants, right? That's the chloroplast. And when this proteobacterium, or a bacteria-like thing, was engulfed, that became our ancestor of animals, fungi, and other heterotrophs. Oh, last question. I keep missing them here. What benefit does the mitochondria or chloroplast get from the cell? So if it has to be mutually beneficial, what do you think they get? Defense? They obviously are within another structure. And that should be... And probably nutrients. That makes sense. So the cell defends the organelles within it from attack and also gathers and provides nutrients for them in exchange for energy. So it's a mutual little handshake between our chloroplasts and the mitochondria. Okay, so let's review mitosis. The process you have seen shows the role of DNA in cellular replication, but there are two organelles that replicate independently. Can you remember them? So we just talked about that. That's the chloroplast and the mitochondria. How do they replicate? By binary fission, the same thing that prokaryotes use. What is the scientific theory that describes why they replicate separately? Endosymbiosis. Okay, so reflecting on what we've learned, binary fission for prokaryotes replicates the DNA, forms two identical cells. Steps of mitosis, can you remember all of them? We start here in interphase, where the cells are mostly hanging out. We go to prophase, the chromosomes condense, the nucleus dissolves. Metaphase, the chromosomes line up at the center line. Anaphase, they start to separate. And then we have telophase, when the nucleus starts to reform around the chromosomes. And cytokinesis, that final step of pinching off the cells into forming two identical cells. What are chromosomes? Chromosomes are, remember we looked at the karyotype. They're all the condensed versions of all the DNA. And they contain genes. And endosymbiosis, the theory that mitochondria and chloroplasts were engulfed by larger cells to form a mutually beneficial relationship called symbiosis, where they exchange nutrients and energy. Cool. So you can take a minute to reflect, and I'll show you that was all one page of notes, and I'll send you a picture of these. And that is what you need to get out of this lesson.